Today we are going to talk about something that we encounter not once, not twice, but several times over in our lifetime. But the process is so commonplace and so mundane in our opinion that we rarely stop to even think about it. As you can see, it is the process of inflammation which forms the first line of defense in our body. And the closely intertwined process with it is that of repair. Inflammation, by definition, is the response of living vascularized tissue to any injury. Now, there are some important conclusions to be drawn from this definition. First is that there needs to be an injury for this response to be mounted. That is the stimulus for the response to be mounted. The next two words that are important in this are vascularized and living. If the injury is of such a nature that it causes the death of the tissue, there won't be any inflammatory response. The second word, vascularized, tells us that for an effective or for an inflammatory response to be mounted, it is imperative that the tissue needs to have an intact blood supply. Again, if the injury is of such a nature that it severs the blood supply, causes the blood supply to be cut off, there won't be any inflammatory response. So remember, it is the response of vascularized living tissue to any injury. Now, like everything, why this inflammatory response? What is the purpose of this inflammatory response? In other words, what are its functions? How does it help our body? So the purpose of this inflammatory response is, number one, to isolate and ward off the injurious agent. Like we saw that any injury is essential for the inflammatory response to be mounted. So the basic purpose of inflammation is to isolate and ward off that injurious agent so that it doesn't cause further damage, and consequently limit the spread of the agent. Then any toxins that have been released can be neutralized by the inflammatory response. And finally, to remove all necrotic cells and tissues that have formed as a result of the injury. And then it is also functional with the process of repair in restoring everything to normalcy. So now let's take a look at the injurious agents that can elicit an inflammatory response. They have been divided into 4 groups for ease of study. Virtually anything can act as an injurious agent, varying from person to person. But these are the 4 broad groups that are important and are usually found at the base of any inflammatory response. First group is that of physical, second is chemicals, third is infective, and fourth is immunological. And as we can see, physical agents can be heat, cold, radiation, trauma. Radiation burns are very severe, very difficult to treat. Extreme heat, extreme cold, they can all produce inflammation. Similarly, chemicals, all organic and inorganic substances can produce inflammatory response if they are applied to the skin, if they are consumed. So they act as poisons, and some poisons are also known to produce the inflammatory response, be it inside the GIT or on the skin by local application. The next group is that of infective agents. They can be bacteria, viruses, and their toxins. So n number of bacteria and number of viruses, they can all be involved in producing inflammation in their host. Then The fourth group is that of immunological agents. They are immune reactions, which can be cell mediated especially, and the antigen antibody reactions. So since we are calling them the enemy, naturally we have and we said that inflammation is the defense mechanism. So there should be some soldiers involved. Let's take a look at what these soldiers are. So as you can see here, the most important part is the blood vessel and especially the blood vessel wall. The endothelial cells, they are integral. Their integrity is breached in different manners to produce the changes seen in inflammation. Then the cellular contents, the intravascular contents, all the cells like neutrophils, lymphocytes, monocytes, eosinophils, basophils, they are all involved, plus the plasma proteins, they are all involved in the inflammatory response. Then you have some extracellular components, the cellular and extracellular, the fibroblasts, the resident macrophages, the mast cells, and finally the connective tissue matrix made up of elastic tissues, the collagen fibers, antiproteoglycans, adhesive glycoproteins, and the basement membrane, which is a specialized component of the ECM or extracellular matrix. So these are all the participants of inflammatory response meant to protect our body initially. Then, like I said initially, we see some signs of inflammation regularly and we just tend to take them for granted. These signs have been summed up nicely long back in the 1st century AD by Celsus, and later on Virchow added one more to it. So now we have 5 cardinal signs of inflammation. The first is rubor or redness. The second is calor, not color. It is calor, derived from calorie. That means heat. Third is dolor, that means pain. Fourth is tumor, that means swelling. And fifth is functiolesa or loss of function. So when there is inflammation, the inflammatory response is mounted. What you see on the surface outside, what the affected person sees is redness, and that area becomes hot, it is swollen up, it is painful, and you are lost to move that organ or do any activity with that limb or whatever is inflamed, that is loss of function. See, this is another depiction of the 5 cardinal signs of inflammation. There is local hypothermia and fever. There is hyperemia or redness, the rubor. Then the tumor is tissue swelling, inflammatory tumors, and there is edema. Dolor is burning pain. This you can see. And finally, the loss of functional or functional impairment is there. We'll see the basis of all this gradually. Inflammation has been traditionally divided into 2, acute and chronic. These are some of the differences between acute and chronic. The first difference that we can see is that acute is of a relatively short duration and is acute in onset, comes up suddenly. Chronic is of a longer duration and persists for very long, lasting for even years. This lasts for acute inflammation lasts for minutes, hours, or even a few days. This chronic can last, like I said, for years together. This chronic inflammation is the cell of chronic inflammation is lymphocyte and also the macrophage. Whereas this acute inflammation is characterized by the presence of neutrophils chiefly. Acute inflammation is exudative in nature and chronic inflammation is proliferative in nature. What does that mean? It means that plasma proteins and cells leak out, exude out of the intravascular system into the extravascular space. So that is exudation. Whereas in chronic inflammation, the characteristic feature is proliferation of the blood vessels and connective tissue to produce granulation tissue. So it is basically proliferative in nature, whereas acute inflammation is exudative in nature. So that brings us to another sideline of this, that we should know what is exudate and what is transudate. This will be dealt with later also in separate chapters, but just a brief overview. Exudate is a fluid that is formed as a result of exudation and is basically a high specific gravity fluid which has high protein content and high cellular content. Whereas a transudate is essentially an ultrafiltrate of the plasma, which is cell poor, has low specific gravity and low protein content. So if you remember that, other finer nuances when we deal with exudate and transudate will be taught in detail. So let's take a look at acute inflammation. Acute inflammation, like I said, is a response to the injury which is immediate. Immediate response to the injury which is going to be short-lived. But it has three major components. Two of them, first and 1 and 2, pertain to the vascular system. So these are known as the vascular events. And third chiefly pertains to the cellular events that take place. Hence, these are known grouped under the rubric of cellular events. So we have two major components, that is the vascular events and the cellular events. So in vascular events, mind you, all these are going on simultaneously. Just for the ease of understanding and study, they have been divided this way. So the vascular events include an alteration in the caliber and subsequently the flow of the system, and also with increased vascular permeability. Whereas the cellular events involve the emigration of leukocytes from circulation to site of injury. So basically, the cellular events involve the journey of the leukocytes from the intravascular space to extravascular space and to reach the site of injury where they carry out their actions. Now, changes in vascular flow and caliber proceed in a very systematic manner. First of all, there is transient vasoconstriction. The vascular system, the arterioles, the capillaries, all constrict in that area initially just for a second or two. And then this is followed by persistent progressive vasodilatation, PPV, persistent progressive vasodilatation. What does that do? It increases the flow, the caliber and the flow. So there is flow of blood, excessive blood in that area. So what happens? There is slowing of the blood flow and ultimately stasis. It is something like a traffic jam. When you are out in the streets and you have a traffic jam, initially the traffic slows down and then finally it comes to a stop. So a similar event takes place here. There is slowing and finally stasis of the blood in that area. Now, normally what happens is that the endothelial cells of the vessel wall are in contact with a cell poor layer of plasma of circulation. But here what happens is that this central axial column containing the cellular element expands. And this causes the margination of the leukocytes so that they come to lie near the endothelial cells. Once they come to lie near the endothelial cells, they start sticking to them. At first, they start sticking transiently, roll along it, and then finally adhere firmly to it. And once they adhere firmly, then they migrate out into the interstitial tissue. So now you can see what I was telling you, that changes in vascular flow and caliber. The vascular events are geared up to begin the cellular events. So it proceeds in a very logical sequence. See, this is the journey of the leukocytes basically, like I said. Initially, they come here. This is the area that has become dilated. So it is coming from here or from here, they reach here. Then they marginate. They have marginated to the periphery. These are the endothelial cells. This is the endothelial cell layer. They come closer to the endothelium. Then they start rolling along it, sticking transiently to it. Then they come to stick firmly here. They have adhered firmly. This is just an example of single leukocyte, but there are rows and rows of leukocytes here. And finally, they are transmigrating out of the vessel into the extravascular compartment. They have reached the extravascular compartment. Now, the second component of vascular events was the increased vascular permeability. And it is the hallmark of acute inflammation, characterized by the escape of protein-rich fluid into the interstitium. So before going on to study how this occurs, we need to take a look at the normal fluid balance. This is dependent on two factors. The first being an intact endothelium. If the endothelium is intact, there is no breach anywhere, the fluid normally will not leak out. There won't be any permeability. And the second is a Starling's hypothesis, which states that there are two types of pressures operative within and outside the vessel wall or the vessel lumen into the extravascular and the intravascular compartments. These two pressures are the colloid osmotic pressure and the hydrostatic pressure. The hydrostatic pressure is, as the name indicates, is due to the fluid. It is due to the fluid. And this causes the movement of fluid from higher to a lower, from lower to higher gradients. And this draws the colloid osmotic is due to the proteins present in the plasma, especially albumin, specifically albumin. And this tends to draw the fluid towards itself. It tends to draw the fluid towards itself. This is a depiction of the normal state of affairs. When everything is normal, there is no inflammation. So this is the arteriolar end, this is the venular end with the capillaries in middle. What happens at the arteriolar end? There is a lot of fluid, so the hydrostatic pressure is 32 mm. And since the proteins are constant, they are not leaking out, staying within the intravascular compartment. It is 25 mm. So there is a net positive pressure of 7 millimeters hydrostatic. So what does it do? It tends to push the fluid outside. Now this fluid has gone out and gradually the pressure is coming down, and at the venular end it comes down to 12 mm. This remains constant. The colloid osmotic pressure remains constant at 25 mm. So now we have a net positive osmotic pressure of 13 mm. And what does it do? It tends to draw the fluid towards itself inwards. So the fluid moves inwards. And whatever little fluid is left behind is drained away by the lymphatics. So that is how the normal balance without any swelling, without any edema is maintained. Now let's take a look at what happens when there is inflammation. Again, the same arteriole, capillary and venular end. Now here we said that there is altered caliber and increased blood flow to that area. So that means increased fluid is coming. So the hydrostatic pressure hypothetically may go up to 50 mm. And this is constant at 25 mm. So there is a net hydrostatic pressure of 25 mm. So it tends to throw the fluid out. And with this fluid going out, the hydrostatic pressure starts coming down. But even finally at the venular end, it may come down to 30 mm. But here there is increased vascular permeability. So this protein rich, the proteins have also passed out a bit. So this has come down to 20, the colloid osmotic. So finally what we have here is 10 mm of hydrostatic pressure positive. Again, the fluid will move out. So at both ends, the fluid is moving out, resulting in edema. So now after having seen all this, we again go back to the 5 cardinal signs, or rather the 4. Fifth is obvious that with all that going on with the 4 events taking place, there's going to be a loss of function. But the first is rubor. There is increased influx of blood in that area leading to redness. Then the calor, again increased amount of fluid will produce redness and heat. Dolor, edema or tumor, the fluid is leaking out, edema is going to be there, so there is going to be a swelling. And all this fluid will press upon the nerve endings, producing pain or dolor. So that explains all the 4 cardinal signs of inflammation, and with all those 4, the natural consequences, loss of function. So that is what you see. So now we have dealt with the normal fluid balance, how it is maintained and what happens during inflammation. And we said initially that normal fluid balance is maintained by an intact endothelium. So during inflammation, there is a change in the endothelium. There is a change in the endothelium which contributes to increased vascular permeability. It has a very important role in increasing the vascular permeability during inflammation, consequently leading to a good inflammatory response. So several mechanisms have been proposed for this, which are the changes which take place in the endothelium to produce this increased vascular permeability. Let's take a look at them one by one. The first is endothelial cell contraction. This widens the intercellular, or specifically the interendothelial junctions. And since it appears immediately and is short-lived, it is known as the immediate transient response. So it is immediate, it is short-lived, that is why it is transient. So it is the immediate transient response. It is seen in mild injury, and it is mediated by some chemical mediators, which are histamine, bradykinin, and leukotrienes. It is reversible, it is short-lived, and affects mainly the venules. Right? So what we have seen is endothelial cell contraction. Contraction means the cells do not move from their space, they just contract their cytoskeleton, and so the intercellular junctions are widened. The next in line, the second mechanism is endothelial cell contraction. This is endothelial cell contraction. It affects venules, it is fast and short-lived, and it leads to widening of the intercellular junctions. The next is endothelial cell retraction. Now what does retraction means? They move back. the endothelial cells move back, one step back. So this occurs because of cytoskeletal and junctional reorganization. This is mediated by mediators like interleukin 1, tumor necrosis factor, and interferon gamma, to name a few. It appears later. It is somewhat later than contraction and slightly longer lived. And it affects both venules and capillaries, but this is a proposed mechanism. They are still uncertain as to how it works in vivo, but still it is a definite phenomenon. Then there is direct injury. Now, if you cause direct injury, the injury is of such a nature that it injures the vessel wall. So there can be two forms of this. It produces its effects in two forms. One is also known as the immediate sustained response. As you can well realize that if the injury is there, there will be an immediate response. And since the gap produced is very large, it will be sustained for a longer period of time. It results, because it results in endothelial cell necrosis and detachment, therefore it will persist for a longer time. So it is known as the immediate sustained response. It is seen in severe injury and causes direct damage. It does not require any mediators. It is just a mechanical injury. It appears fast, lives long, and affects all levels of the circulation, that is venules, arterioles, and capillaries. The second part of this, the direct injury, can also produce a delayed prolonged leakage. It can also produce delayed prolonged leakage. It is known as delayed prolonged leakage, means it appears after some time. It begins after a delay of 2 to 12 hours, but it lasts for hours to days. That is why it is known as the delayed prolonged leakage. It is seen in mild to moderate injury. The possible mechanism that has been proposed for this delayed prolonged leakage is that first is via apoptosis of the injured cells. They undergo apoptosis, so that naturally takes a bit of time. And the second is via cytokines. The cytokines that we talked about in the contraction and retraction. So these cytokines, they lead to retraction. So it has a roundabout way producing retraction. So these are the two proposed mechanisms of how delayed prolonged leakage occurs. It affects venules and capillaries chiefly. Keep in mind that these are the main areas affected, but these can affect any level of circulation. See, direct injury, this is the leakage that is happening. This is the endothelial cell. This is the intravascular compartment. This is the extravascular compartment. So this is injured. This is normal cell. So fluid is passing out. Then the fourth mechanism that has been proposed is leukocyte-mediated endothelial injury. As we saw that the leukocytes were adhering to the endothelium, and this movement of the cells and adhesion is all collectively known as activation of the lymphocytes. This movement is one part of activation. There are several chemicals which are released. They are toxic oxygen species and proteolytic enzymes. So what do these enzymes do? When they are released outside, they cause damage to the surrounding tissues and especially the endothelial cells. So they cause damage to them, they become detached. So this appears once they get activated, release the oxygen species, so it takes a bit of time. So this appears late, but again, this repair will also take some time. So this mechanism, the leakage by this mechanism is long-lived. It affects chiefly the venules. So this leukocyte-mediated endothelial injury appears late, but is longer-lived. The next mechanism that has been proposed, this is the graphic of this. These are sticking to this endothelial and producing damage here and here. The next is leakage from regenerating capillaries. During repair, angiogenesis takes place. Angiogenesis means the formation of new vessels. They are formed by proliferation of the endothelial cells. So naturally, we saw in the beginning that the endothelial cells have interendothelial junctions. Two endothelial cells have a junction which is firm and does not allow any leakage. But when new vessels are formed, the endothelial junctions are not properly matured. So when proper differentiation takes place and the junctions become firm, then the leakage stops. But prior to that, leakage continues, and that is one mechanism which has been proposed for increased vascular permeability during inflammation and repair. And the next or the last one, this is, see, this repair is going on, this is immature, junction is not there. Here also junction is not there. These two are mature, so there is the junction is intact, no leakage. Next is increased transcytosis. This is the last mechanism that has been proposed. There is a vesiculo-vacuolar system across the cytoplasm in all cells. And there is movement constantly of fluid inside that. But this can, once it crosses the cell boundaries, then the fluid leaks out. And this is mostly seen in blood vessels within tumors, where again the cells are not properly formed. They are proliferating at a higher rate. So the vesiculo-vacuolar system across several cytoplasmic compartments will take place and result in increased vascular permeability. This is how it occurs. This is the endothelial cell. This is the vesiculo-vacuolar system, and this becomes active and causes leakage. Openings and leakage will be there. This is communicating to the surface. This is communicating to the surface. So across the cells, it is taking place. So to summarize, we have seen that there is contraction, retraction, direct injury in the form of A and B, the immediate sustained response, and the delayed prolonged leakage, and then the leukocyte mediated injury, the regenerating capillaries, and transcytosis. So this summarizes how the increased vascular permeability in what ways can contribute to the leakage of protein-rich fluid into the extravascular compartment. The second component of inflammation was the cellular events. The first were the vascular events, which we have dealt with. Now the second is the cellular events that take place. So important function of inflammation is to see that the leukocytes reach the site of injury. So we have already typed a bit about it. Now we'll go into the details. It is, like I said, the travelogue or detail about the journey of the leukocytes from the intravascular compartment to extravascular compartment to reach the site of injury and bring about the effect of killing or degradation of the bacteria. So what happens in the lumen inside the blood vessel? First, there is margination, which we have seen, rolling and adhesion. Firm adhesion can, after that, there is across the endothelium, there is transmigration. And then finally, when they come to lie in the interstitial tissues, there is migration toward the chemotactic stimulus to reach the site of injury. They essentially do not come out at the exact site. They come out in the near vicinity, and then some chemical mediators help them to move along a chemical gradient to reach the site of injury. So this is the journey of leukocytes, which constitutes the cellular events of inflammation. And as you can very well appreciate that there is a lot of overlap in the cellular and the vascular events of inflammation. Now, as I typed briefly before also, that normally RBCs and WBCs occupy the central axial column, and a cell-poor layer of plasma is in contact with the endothelium. Now, in inflammation, what happens? Due to the slowing stasis, first influx of the red cells and the blood into that area, the cells fall out, the central column expands, and the cells tend to fall out of the central column and occupy a peripheral position along the endothelial surface. This is known as margination. The cells occupy the margins. Next was, see, this is, they tumble and reach here. They are marginated along the endothelium. This is known as margination. The next step was rolling. First single cells, then rows of WBC tumble slowly along the endothelial surface, adhering transiently to it. They stick there for a brief moment, then tumble along more, then stick, then tumble along more, and finally, at some stage, they come to stop. This is known as pavementing. Why? Because ultimately, all the cells line the layer of the endothelium so that you see a full layer of endothelial cell covered by a layer of neutrophils. This is somewhat similar to the appearance as you see at the base of a swimming pool, which is covered by tiles. So the leukocytes are lining that, and whatever water is flowing above it, that does not disturb the tiles. So similarly, the cells are not disturbed by the flow of plasma, which goes on uninterrupted, and these adhere to the endothelium. This is known as adhesion. And finally, there is transmigration. Following the firm adhesion, now what they do, they insert their pseudopods between the endothelial cells and squeeze out through the interendothelial junctions, which we have already been talking about. And then they position themselves between the endothelial cells and the basement membrane of the blood vessel. And then they secrete collagenases, which creates an opening for them in the basement membrane, and they escape into the extravascular space or the ECM of the extracellular matrix. See, this is inserting out, this cell is coming out, throwing a pseudopod. This is the higher view, and this is a graphic depiction. It is coming out through the blood vessel wall. Now, the question comes to our mind, or it should come to our mind, that what happens, why they start sticking to it, why the cells start sticking to endothelium. Do some changes take place? So they definitely do. Since this is known as adhesion, the molecules that bring about this, the molecules that are responsible for this change have been named as the adhesion molecules. They bring about adhesion, and they are present both on the endothelial cells as well as on the leukocytes. They are present both on the adhesion molecules on the leukocytes as well as on the endothelial cell surface. In some cases, they are newly synthesized. In some cases, they come to the surface where while they were lying previously inside, some sequestered inside the viable pellet bodies, as we'll see. So the ultimate result is that they are present, appear on the endothelial surface, and they appear on the leukocytes. And both of them are complementary to each other, and they stick to each other and bring about the firm adhesion and later on the transmigration. So what happens? The adhesion receptors or molecules belong to 4 families. There are several of them, but we are mainly concerned with these. The first group is of selectins, second group is the immunoglobulin superfamily, the third are the integrins, and fourth are the mucin-like glycoproteins. Now, just by the way, I'll tell you that the selectins and mucin-like glycoproteins are complementary to each other chiefly, and immunoglobulins and integrins are complementary to each other. But let's take a look at the selectins. They are the E, P, and L selectin. Some of them, sometimes they represent the names. E is from the endothelium, L is they appear on the leukocytes. So that is how it goes on. The immunoglobulin superfamily consists of molecules like VCAM1, vascular cell adhesion molecule, ICAM1, there's another that is PCAM1. So they are being routinely added, more and more are being discovered. So this, then we have the integrins, which are beta 1 and beta 2 integrins. Beta 1 are VLA4, LFA, and beta 2 are MAC1. So, and then we have the mucin-like glycoproteins. So let's take a look. Like I said, the first and the fourth group are complementary to each other. That is mucin-like glycoproteins and the selectins. Now the selectins have an N-terminal domain or the lectin domain. Now this N-terminal domain or lectin domain binds to a silylated oligosaccharide like silyl Lewis X, for example. Now this oligosaccharide, the silyl oligosaccharide, is already bound to the mucin-like glycoprotein. So something like this selectin through its N-terminal domain, binds to the silylated oligosaccharide, which is already bound to mucin-like glycoprotein. So it is through this that these complementary molecules bind to each other and cause the sticking of the leukocyte to the endothelial surface. Then the 2 and 3 were complementary, the immunoglobulin superfamily and the integrins. How? The ICAM and VCAM are present on the endothelium, whereas integrins are present on the cells. So now the beta 2 group binds with ICAM1 and the beta 1 binds with VCAM1. Again, the cells and the endothelium come in contact with each other through these adhesion molecules and are bound firmly to the surface of endothelial cells. Now, like we said, what happens to these molecules? Normally, they do not bind. Normally, they are not even seen. Normally, they have no action. So what happens during inflammation? So how the adhesion is induced, how it takes place, how it begins. So first mechanism that has been proposed is that there is redistribution of adhesion molecules to cell surface. Some of the molecules are already present inside the cell, they come to lie on the cell surface. The second is induction of adhesion molecules on the endothelium. They are being synthesized on the adhesion molecules, and sometimes they are at low level of synthesis of numbers, more numbers are there, increased expression. And then in some cases, there is increased avidity of binding. It is not necessary that there will be a single mechanism operative. There can be more than one mechanism operative to bring about the addition of the cells to endothelium. Now, taking a look at redistribution, we find this is in case of P-selectin, the first group, the selectins, E, P, L selectins. Out of that, the P-selectin is an example. There is some bodies known as the Weibel pellet bodies, which are present in the endothelium, in the some intracytoplasmic granules, which are known as the Weibel pellet bodies, which are present in the cytoplasm of the endothelium. They contain P-selectin. Now, once there is stimulation by histamine, thrombin, platelet activating factors, whatever is the stimulus, the P-selectins get redistributed to the surface and binds the leukocytes. This phenomena may be important in rolling. See, this is a depiction. These are the Weibel pellet body, and this is the P-selectin within the cyto. This is the endothelial cell, Now, once there is stimulation by histamine and thrombin, they migrate from here and come to lie on the surface of the endothelial cell. Now, here they are capable of binding the leukocyte with the complementary molecules, which we already saw was mucin-like glycoprotein. Now, increased induction and increased expression. The second mechanism that we saw was, you can take an example of E-selectin. This is normally absent on the endothelium. Once there is secretion of interleukin-1 and tumor necrosis factor, then there is induction or synthesis of the E-selectin after a gap of 4 to 6 hours. Right? So they are maximally seen on the surface of endothelium after 4 to 6 hours, the E-selectins. This will mediate adhesion of neutrophils. Since this is the second step, see how systematically nature proceeds. First, the P-selectin came into play because it was important in rolling. And now we need addition. So after 4 to 6 hours, these are maximally induced and they mediate the addition of neutrophils. Now, another increased expression was that VCAM and ICAM, they are normally present at low levels on endothelium. The cytokines, once they are released, they increase their expression. So more VCAM and ICAM is present on the surface. For availability, the E-selectin is newly synthesized. These are maximally increased in expression. See, the E-selectin or ICAM1, we take the example. This is the neutrophil which is activated and is showing its addition molecule. This endothelium has nothing now at present. Once there is stimulation by any cytokine, interleukin-1, tumor necrosis, this, either it is E-selectin or ICAM, depending on whatever stimulation has been, they are expressed maximally on the surface of endothelium cell. Either they are synthesized newly or they have been increased in expression and they bind these adhesion molecules on the leukocytes and bring about adhesion. From rolling, they are passed on to adhesion. Then increased avidity of binding. In this, we'll take an example of integrin LFA1. It is normally present on the neutrophils and cells, all the monocytes and lymphocytes, and does not adhere to its complementary molecule as we saw ICAM1 on the endothelium. Normally, it does not. But once there is activation of the leukocyte through any mechanism, any stimulus, the LFA1 goes up to a state of high affinity binding towards ICAM1. And as we saw in the previous slide, it is already ICAM1 is showing increased expression. So this results in firm adhesion. Firm adhesion. And once this firm adhesion takes place, the next step is that of transmigration. That transmigration is mediated via PCAM1. Now there can be some defects in this finely orchestrated chain of events. So it can lead to impaired leukocyte adhesion and subsequently impaired mounting of the inflammatory response and subsequently to recurrent infections. This is known as, some of them which have been documented are LAD type 1, leukocyte adhesion deficiency type 1, in which there is a beta 2 chain defect. LAD type 2, in which there is absent silyl Lewis X, so no glycoprotein and selectin binding will take place, so there will be no rolling. Then antibodies to PCAM1, they affect transmigration because PCAM1 is involved in the transmigratory process. Now once they come to lie outside, they have to move along a chemical gradient. So that is known as chemotaxis. So chemotaxis is known as the locomotion along a chemical gradient. It is sort of a guide for the cell to reach the proper, something like a Google map. You will identify more with that. So something like a Google map which shows the direction to the cells where to reach and how to reach. So they reach the site of injury after transmigration with the help of the process known as chemotaxis. So naturally for chemotaxis, you need some chemoattractants which will attract the leukocytes towards the site. So they have to be present at the site of injury. They can be of two types, exogenous and endogenous. Exogenous are those that come from outside. They are most commonly the bacterial products, the injurious agents that have entered and have been broken down, the bacterial, or they have secreted some toxins, something which will act as chemoattractants, and they are the bacterial products. The endogenous are, they are also numerous, but to name a few, they are C5A, that is the activated form of complement number 5. Leukotriene B4, which is a product of arachidonic acid metabolism, and the interleukins are also secreted by the various cells. They are the interleukin 8 family. So these act as chemoattractants, causing the cells to move along that chemical gradient. Now, there are several receptors present on the surface of leukocytes. They may be toll-like receptors, they are G protein coupled receptors, they are receptors for rolling, they are receptors for adhesion. So, but the basic process which is comes under the rubric of activation of leukocyte remains the same. Whichever receptor for killing, receptor for binding, whichever receptor binds to the proper chemotactic agent and activates the neutrophil. So what happens? We'll take an example of G protein coupled receptor here. It causes activation of the phospholipase C, which by the process of hydrolysis causes conversion of phosphatidyl inositol biphosphate to DAG and diacylglycerol and inositol triphosphate. Now this IP3, this is very important. It increases the calcium. It causes intracellular increase in calcium by drawing calcium from outside. And this is responsible for production of the changing the structure of the cytoskeletal rearrangement and causing more locomotion. The pseudopods are produced and causing the leukocyte to move along the gradient. So this is the most important action that we are concerned with today. But the other actions that also take place along with this, that DAG was produced, it causes protein kinase C activation, which leads to phosphorylation of the granules, which are present in the neutrophils through process of phosphorylation. It results in the oxidative burst, degranulation, and secretion of the chemicals inside, which results in killing of the bacteria as well as in producing the tissue damage inherent in the process of inflammation. Innocent bystanders are also harmed. The via lipase, again with calcium, DAG acts on phospholipids which are present in the cell membrane. This produces arachidonic acid with the help of phospholipase A2. Again, arachidonic acid metabolism is an important integral part which produces several cytokines and several thromboxane A2, all are produced which help in the process of inflammation. Now, like I said, it is most important for us to see the release of calcium intra and extracellularly leads to locomotion and other responses. At present, we were talking about locomotion only. So see, this is a diagram showing the important receptors here. This is the G protein coupled receptor, which we discussed. This is the toll-like receptor. This is again the cytokine receptor. This is again the phagocytic receptor. So depending on whichever is bound, usually all get bound and produce activation. So the recognition of microbes and mediators is through these receptors. Okay, once they are recognized, they produce a cellular response within the cell. Sometimes there is cytoskeletal changes, so this will produce chemotaxis and movement, production of other mediators, production of reactive species, and phagocytosis. Finally, it will result in addition to endothelium, activated lymphocyte, neutrophil, migration into tissues, transmigration we have talked about, amplification of inflammatory response through arachidonic acid metabolites, and killing of the microbes. So these are all the actions, but we were concentrating on the chemotaxis or the movement and migration. But these all will take place simultaneously during the process of inflammation. These are the functional outcomes of the activation of a neutrophil. Now, this is the most important aim of inflammation, the killing and degradation, and before that they have to engulf the offending agent, and that is through the process of phagocytosis. So first, before phagocytosing the material, the cell has to know what is the offending agent, what it has to engulf. So for that, nature has provided a mechanism, that is the opsonization. That is recognition and attachment of the particle to be ingested. After that, engulfment and formation of a phagocytic vacuole, and finally killing or degradation. These are the three steps that take place, complete the process of phagocytosis. So the first step is recognition and attachment. There are certain substances which are known as opsonins. Loosely translated, opsonins are some morsels that make the food more tasty. Literal explanation is, so they render the bacterial product to be attached, more tasty, or more recognizable, so that the cells can recognize them. They are Fc fragment of IgG. Corresponding receptor is Fc gamma R. Then C3b and C3bi, the complement fractions of complement 3. CR1, 2, 3 are the corresponding complement receptors. Then the collectins and the C1q is the corresponding receptor. Now opsonins coat the bacterial products, and then they bind to these receptors on the cells. Now the engulfment. Once they have been opsonized, identified, rendered more tasty, now the leukocyte knows that it has to engulf them. It reaches there, and the pseudopods flow around the object and completely enclose the particle, which is known as the phagosome. Now this fuses with the lysosomal granules present in the cell and is now known as the phagolysosome. Now lysosome discharges its contents in the phagolysosome and causes, this is known as degranulation. The granules are discharging their contents. And finally, the ultimate step for which all this was going on, the killing or degradation of the bacteria. This can be by through two mechanisms. One is oxygen dependent mechanism, and the second is oxygen independent mechanism. The oxygen dependent mechanism, there is a burst in the oxygen consumption, which results in glycogenolysis, and there is increased glucose oxidation. So this leads to the production of reactive oxygen metabolites. Now this oxygen metabolites, along with our activated oxidase, acts upon this with NADPH to produce the toxic oxygen radicals and hydrogen molecules. This, along with this oxygen radicals and hydrogen molecules, undergo spontaneous dismutation to produce hydrogen peroxide or H2O2. Now this H2O2, via two pathways. The Fenton reaction or the Haber-Weiss reaction produces hydroxyl ion, which is a toxic ion and produces destruction. Number one. Number two, it takes up the MPO from the cell, the myeloperoxidase. Hydrogen peroxide is acted upon by myeloperoxidase plus one halogen. In this case, we are taking the example of chloride. It can be bromide, it can be nitride, anything. One halogen is taken. Results in the formation of hypohalous acid. In this case, hypochlorous acid. This hydrogen peroxide NPO the halogen method is the most powerful bactericidal system in the cell. Then we have the oxygen independent mechanism. These are present again in the leukocytic granules, some substances which produce the destruction of the bacteria. Like, for example, BPIP, bacterial permeability increasing protein, bactericidal permeability increasing protein. This increases the permeability of the membrane of the bacteria. Then we have the lysozyme, which chiefly acts through hydrolytic actions. Then we have the MBP or the major basic protein, which is mostly cytotoxic to many parasites. Therefore, it is produced naturally by eosinophils. That is why in parasitic infections, you see a preponderance of the eosinophils in the parasitic inflammations. Then defenses again produce holes in the membranes of the bacteria. Then once the microbes are killed, they were killed by the oxygen dependent or the oxygen independent mechanism. Now the lysosomal acid hydrolases come into action. They degrade the bacteria within the phagolysosome. The killed bacteria are now, they are kept sequestered because the lysosomal enzymes are very powerful and they can produce damage. They do also, but mostly they are kept sequestered within the phagolysosome. Then these kill the bacteria, but whatever products are released outside inadvertently, they cause endothelial cell injury and tissue damage as well. So that is all for today. Next turn, we'll take up chemical mediators of inflammation.